So our last speaker is the Medical Student Summer Research Foundation Award winner, and this topic is dose-dependent response of neuroinflammation after repetitive mild traumatic brain injury, and the speaker is Stephen Miranda.
Thanks for sticking with me here. So my name's Stephen Miranda. I'm a second-year medical student at the University of Rochester. And first I'd like to start by thanking the AANS for sponsoring my research last summer and giving me the opportunity to present it here today. So I have no disclosures to make. And so many studies recently have suggested that repetitive mild traumatic brain injury might be implicated in chronic neurodegenerative diseases like CTE, chronic traumatic encephalopathy. And now, if this is indeed the case, it would become important to understand when patients might be at risk for progressing to neurodegeneration. And so as a result, we were interested in exploring that relationship between repetitive head injury and the onset of symptoms. And we were wondering specifically, might there be a threshold number of hits to the head, at which point a pathologic process might be initiated? And so there are a number of variables that affect repetitive head injury, particularly the timing in between hits, the number of hits, the severity of those hits. But we were specifically wondering, we wanted to assess whether there might be a dose-response relationship between the number of head impacts and the onset of any neuroinflammation or behavioral changes in mice. And so to test this, we delivered increasing numbers of hits to different groups of mice, but we gave those hits over the same time period to replicate one football season or one military tour of duty. And we predicted that mice that received more hits over this time period might experience greater degrees of neuroinflammation and maybe possibly more profound behavioral changes as well. So in our experiments, we modified our previously published hit-and-run model. And so we made three specific changes to more accurately replicate the biophysics of a head impact. Firstly, the pneumatic piston used to deliver the head impact was covered with a rubberized tip to more broadly distribute the force over the left temporoparietal lobe. Secondly, the mice were not subjected to anesthesia. Anesthesia has been shown to be neuroprotective, and in some instances even neurodegenerative by promoting hyperphosphorylation of tau protein. So we wanted to avoid those confounding factors as well. And lastly, the mice were allowed to swing freely upon impact to introduce a rotational component of injury in addition to the acceleration-deceleration component of injury that the brain might experience. So with this modified hit-and-run model, we created 4 experimental groups, receiving 5 hits, 15 hits, 30 hits, and 60 hits over a 5-day period. And then 3 days after the last impacts were delivered, we assessed behavior in these animals, specifically with regards to motor function, depression, and cognition and risk-taking. So going straight to the results, on the left-hand side, we assessed motor function with the open field test, where the mice were allowed to roam over an open enclosed space. And as you can see here, there were no significant differences between the experimental groups with regards to the total distance at which they traveled and the mean speed at which they traveled, suggesting that their motor function was pretty much intact. We then looked at depression using 2 validated tests for depression in mice, the Parsalt Forest Swim test and the tail hang test. Now, in both of these tests, when a mouse is suspended by its tail or is placed in water, the more time the mouse spends immobile, the more depressed that mouse is said to be. And so looking here at the bottom of the tail hang test, you can see that our 30-hit mice spend significantly more time compared to controls immobile, as did our 60-hit mice. And looking at the forest swim as well, the 30-hit and 60-hit mice spend significantly more time compared with controls and the other experimental groups immobile, indicating a degree of depression in these mice. We then assessed cognition with the novel object recognition test. Now in this test, when a mouse is confronted with a novel object and a familiar object, it tends to spend more of its time with the novel object. However, if there's any cognitive impairment or any memory problems, the mouse might have difficulty remembering the familiar object and might have trouble distinguishing between the two and might spend less time with the novel object. So as you can see here, our 60-hit mice spend significantly less time with the novel object compared with the other experimental groups. In fact, they spend close to 50% of their time, which is virtually the same as chance, or not being able to distinguish between the two objects at all. So we noticed some cognitive impairment in our 60-hit mice. We then, in the top right, we then assessed risk-taking behavior with the elevated plus maze test. Now in this test, there are 2 enclosed arms of the plus, and the other 2 arms are open. And a mouse will normally like to spend its time in these safe, enclosed arms. But as you can see, our 60-hit mice spend significantly more time in the open arms, which is a bit daring, demonstrating some risk-taking behavior. In fact, our 60-hit mice spend almost 80% of their time in the open arms, which is much more than controls and significantly more than even the 5-hit and the 15-hit mice. So taken together, these results show that the more hits the mice received, they seemed to be more depressed, had worse cognition, and demonstrated some increased risk-taking behavior. However, their motor function remained largely intact. So moving on, we wanted to assess if there might be any neuroinflammation in the brains of these mice that might correlate with these behavioral deficits. So we did this by measuring GFAP expression. And GFAP has been validated as a marker of reactive astrocytosis. And so on immunohistochemistry, we looked at these as fluorescence to measure GFAP expression. And then from these representative images, you can see that as you get up to 15 hits, 30 hits, and 60 hits, there appeared to be more GFAP expression, indicating more neuroinflammation. However, we wanted to quantitate this, so we expressed the amount of fluorescence that we saw using image analysis software as a percentage of the total area of the brain sections. And then comparing this fractional area of GFAP expression, we could compare the amount of neuroinflammation across the cortex, subcortex, white matter, and the hippocampus. So looking at these results, we can notice three interesting things. Firstly, there appeared to exist a dose-response relationship between the number of hits the mice received and the degree of astrogliosis in their brains. And this trend was consistent across all four areas. With the more hits the mouse got, the higher the amount of GFAP expression. Secondly, and more interestingly, there appeared to exist a threshold number of hits at which this astrogliosis became significant. So for instance, if you look here at the subcortex, our 5-hit mice displayed no significant differences compared to controls with regards to GFAP expression. But as you get to 15 hits, this difference suddenly becomes significant, and as you increase the number of hits, the amount of GFAP expression goes up as well. And this was seen across all four regions. Thirdly, I just wanted to point out to the hippocampus as a unique case of initially at baseline there appeared to be more GFAP expression. And then when you get to that 15-hit threshold, there's a marked elevation in GFAP. However, it seems to plateau as you go up instead of increasing in a linear dose-response fashion like we saw elsewhere. You might remember from our novel object recognition test that our 60-hit mice had possibly some memory impairment as they had trouble distinguishing between the two objects. And interestingly, even patients with chronic neurodegeneration, like CTE patients, also have profound memory deficits. So it makes sense that the hippocampus might be uniquely implicated, but we can't fully explain why the curve is the way it is here. Nevertheless, we can draw some significant conclusions from these data. Firstly, the number of head impacts that we delivered to the mice appear to correlate very closely with the extent of observed behavioral deficits in these mice, including increased depression, worse cognition, and increased risk-taking. Secondly, the number of hits also appear to correlate in a dose-response fashion with the degree of astrogliosis in the brains of these mice. And interestingly, we noticed a threshold number of hits consistent across all brain areas, at which point the astrogliosis became significant. Now, what might these findings mean in a broader clinical context? I mean, granted, we did assess behavior at 3 days out, and with regards to neuroinflammation, we did assess it in a relatively acute setting. However, if these findings were to be confirmed in humans, it could have great clinical significance in the sense that we could prevent patients from reaching that threshold number of hits and prevent them from going on to chronic neurodegeneration. But we still have a long way to go with this work to get to that point. And there are a number of future directions we can go. Firstly, we could follow these mice out long term and see what happens to them in terms of their behavior, as well as any pathology. Secondly, what if we changed the hit frequency? We delivered all those hits over the same time period. And what if we spread those hits out over a longer period of time, decreasing that frequency? Would that ablate the histology we see or change the behavior we notice? Additionally, what if we use a different mechanism of TBI delivery, such as blast TBI, which we've heard about today? How would that change things? And lastly, what exactly is going on at this threshold number of hits in terms of a pathologic mechanism? When a certain number of hits is delivered, does that change solute or waste clearance, leading to more inflammation and possible behavior changes? That really is what we should try and understand. I'd just like to end my talk by thanking Dr. Edward Vates, my AANS mentor who sponsored me for this fellowship, Dr. Macon Niedergaard, whose lab and resources supported my work, and my collaborators, Dr. Matthew Dashnaw and Benjamin Plog. Thank you very much, and if you have any questions. Hi.
Hi. Congratulations on really outstanding work. I think this is the kind of basic science that we need to start doing to address the question of CTE. As you're probably well aware, CTE is characterized by a tauopathy. So my question for you is, have you looked at tau staining in your model, or do you plan to do that at some point in the future?
So yeah, absolutely. We do plan to do that in the future, but we haven't looked at the pathology in these mice with regards to tau protein, phosphorylated tau. But that would definitely be a future direction.
Is there a change in the axonal injury in these brains? Like, you've looked at the astrogliosis. Have you actually looked at the axons and stuff?
No, actually, I have not looked at the axon. I mean, it'd be interesting to see if there's any diffuse axonal injury as well. But I only had time for in the summer, just GFAP expression, but that would be a worthwhile future direction as well. Thank you very much.
So if there's no other questions, then I'd like to close our session on neurotrauma and critical care. And thank you all very much for sticking around to the end.